Hello, everyone. Thank you all for joining us today. This is our first webinar in the new EPIC webinar series on the use of NOMs in risk assessment. The series is being co-organized by the Environmental Protection Agency, the PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. We're so happy that you're all joining us today. We had lots of people sign up, so I'm going to give a second to let everyone log in. But I'll introduce myself. I'm Bridget Rogers. I'm an advisor to the PETA Science Consortium, and I will be co-moderating today's webinar along with several others, including Rachel Bruner, a biologist, and Gino Scarano, a senior advisor, both from the New Chemicals Division of the EPA Office of Pollution Prevention and Toxics, Naladri Baumick, a toxicologist on the Human Health Assessment Branch of the California DPR, and Christy Sullivan, the Vice President of Education and Outreach at IIVS. Okay, I will turn it over to Rachel now. Rachel?
Thank you. So we have two speakers today who will be presenting on OECD updates and the Developmental Neurotoxicity In Vitro Test Battery. First, we'll hear from Charles Kovatch with the EPA Office of Chemical Safety and Pollution Prevention. And then we will hear from Tim Schaefer from the EPA Office of Research and Development. Before I introduce our first speaker, note that the slides and a recording from today's webinar will be posted online at this link shortly after the webinar. All future EPIC webinar materials will also be available at this link as the series continues. Please note the presenters' cameras will be off during their presentation to preserve the sound quality for the recording, but they will be on screen for the questions that we will take at the end of the second presentation. Everyone is on mute, but you can type any questions into the questions section within the GoToWebinar toolbar, which should be on the right side of your screen. And please indicate if your question is directed to a specific speaker. So now Gino Scarano will share some introductory comments from the EPA.
Thank you, Rachel. It's my pleasure to welcome the speaker, the panelists, organizers, and all of you joining today for this, our first EPIC webinar. This one's on two topics, OECD updates by Charles, and Tim Schaefer will talk about the DNT, or Developmental Neurotox Test Battery. Allow me to be nostalgic for just 1 minute, please. So I want to say goodbye to the PEP webinars. So as some of you may know, PETA, the International Science Consortium, EPA, and Christy, when she was with the PCRM, Physicians Committee for Responsible Medicine, we co-hosted 12 webinars between 2018 and 23. But as we say goodbye to the PEP webinars, the vision lives on in this new EPIC webinar series. So I'm really excited. And it's the same vision, bringing timely topics on NAMs and their use by regulators and in risk assessment as we try to advance the use of NAMs. I'd also like to specifically welcome two new people to our planning committee, Naladri Baumick from California DPR and our own Rachel Bruner in the New Chemicals Division at EPA. So it's exciting to have this new blood and help on the planning team. And as always, we are indebted to PETA for doing all the logistics for this, which is very helpful, and finding a home for the recording and the slides, both for the old PEP webinars as well as these new EPIC webinar recordings, which will be posted online. With that, I'm looking forward to hearing both Charles and Tim, and I give the floor back to you, Rachel. Thank you.
Thank you, Gino. I'd next like to introduce our colleague from the California Department of Pesticide Regulation, Naladri Baumick.
Thank you, Rachel. And first and foremost, I'd like to share an exciting news. India today, for the first time all over the world, made a soft landing on the South Pole of Moon just a little while ago. So it's exciting. I'm in India right now, so I can feel the excitement here. And thank you all panelists, co-organizers, organizers for including California Department of Pesticide Regulation. And we are super excited to be part of this very useful webinar series on new approach methodologies. Thank you, and back to you, Rachel.
Thank you, Niladri. Okay, so speaking first today will be Mr. Charles Kovatch. Charles is a senior scientist with the EPA Office of Chemical Safety and Pollution Prevention. Since 2021, he has served as the U.S. government's national coordinator to the Organization for Economic Cooperation and Development, or OECD, test guideline program. In this role, he coordinates among the U.S. federal agency partners to communicate the U.S. government's priorities to OECD in the development of test guidelines. Mr. Kovatch has over 20 years of experience working with federal, state, tribal, and local agencies, as well as industry and associations on national policy issues. He has also performed details to the U.S. Senate as a Legis Fellow and to the White House Council on Environmental Quality. Mr. Kovatch has a bachelor's in environmental health sciences from Indiana University of Pennsylvania, a master's of public health in environmental toxicology from the University of South Carolina Arnold School of Public Health, and an MBA from the University of Maryland Smith School of Business. Welcome, Charles, and please go ahead.
Great, thank you. Okay. Guess I would ask for my first slide to be, there it is, great. Well, thank you again for this opportunity. I'm happy to be here today and serve as the first speaker on this series. With that, next slide, please. So today I'll provide an overview of the WNT meeting that was held back in April, talk about some of the other program updates and a few upcoming activities. Next slide, please. I know we have a diverse audience today, and so I thought it'd be nice to start and take a step back to revisit why we're here. So regulatory agencies are tasked with making decisions on whether a chemical, whether from commercial industrial uses, pesticides, biocides, cosmetics, pharmaceuticals, or agrochemical, or nanomaterials, or other, are safe for use to human health and the environment. And so, there are always questions about, well, what methods or protocols were being used to test this chemical, or how do I even interpret this new technology that's now coming in the form of these new assays, or how can I even trust the data that's behind this study that's been handed to me? So with that, next slide, please. The OECD since 1961 has been bringing together now 38 member countries and other partners to collaborate on global economic, social, and scientific issues on a range of areas. And this is just a few of them. I have listed here like tax policy and digital transformation and climate change, energy, trade, education, finance, and others. The area where we work is the Chemicals and Biotechnology Committee, or the CBC. The CBC oversees work on chemical safety through the activities on 12 different working parties that include hazard assessment, pesticides, nanomaterials, and the WNT, which is the test guideline program. The CBC has established the MAD system, or the mutual acceptance of data, to provide a common level playing field for cooperation among national authorities across the OECD member countries and other countries that adhere to MAD, even though they're not a member. So MAD establishes sound science through the test guidelines, and you could say study quality through the good laboratory practice program to provide trusted and accepted data so that regulatory authorities receiving the data under MAD know that there's a particular quality and scientific standards followed for that study. It also means that companies can do a single test and then submit that data to regulators in multiple countries rather than undertaking separate tests to meet different requirements. So I would also just caveat that acceptance doesn't mean use. So that decision still does fall to every individual government regulatory decision. So lastly, on this slide, the test guideline program. So I'm the US representative. I'm also have a co-lead, Nicole Kleinstruer, who is the director of NICEDM at the National Institute of Environmental Health Sciences. So together, we represent the US with the other member countries and other MAT adhering countries to harmonize and improve test guidelines in accordance with 3 Rs. And so with that, next slide, please. All right, so this is an oversimplified diagram of the test guideline development process. However, I think it's helpful just to at least provide you with an overview of the process and serve as reference points for the remainder of this talk. So it also wants to convey, I also want to convey that there's a lot of work to develop an idea, demonstrate repeatability and validity, have it reviewed by experts, and then approved by the WNT. And so these steps, like proposing, the first one is, develop an standard submission form, what the WNT uses, the SPSF. So develop that proposal for a new method, or maybe revise an existing method, and identify a context of use. And once that proposal has been reviewed, the WNT will make a decision on whether to approve it and then add it to the work plan. Then the next step for a project would be to work on selecting the reference chemicals, standardizing their procedures, developing the SOPs, determining different data interpretation procedures, identifying partner labs and conducting inter-lab testing, and then developing a validation report, that would, talk about domain of applicability and different limitations of the proposed approach. And then you can develop a draft test guideline, right? Or make revisions on an existing one. Once you have those draft validation reports and test guidelines, those are reviewed by the expert groups and other mechanisms. Once those are in a place where they feel they're in a good shape, then they come to the WNT for another round of commenting among the national coordinators, and then a decision on their approval. And then once approved by the WNT, the CBC, which is, of course, again, the governing structure, will make their final determination on approval and then publication. So it's a long process to reach consensus, to have a product that can take 1 year or up to many years. Next slide, please. So I'll talk about these in greater detail, but the WNT meeting, so remember we talked about the meeting that was held recently in April, we added 12 new projects, the SPSFs, that were now placed on the WNT work plan. And then we approved 10 items as test guidelines or guidance documents or detailed review reports. And then the US, I'll be talking about all these, that the US led and co-led some activities on proposals and test guidelines. We participated in almost every project that was approved. And then we will likely be participating in some capacity on any of the new SPSF projects, and then some other updates. So next slide, please. This is a list of all the project proposals which were approved, and I want to make sure that we're not going to talk about all of them, but really just to highlight 2 of them where the US will be the lead or the co-lead. One is the detailed review paper, so like literature review, if you will, to facilitate the development of test methods to predict the respiratory sensitization potential of substances. And so again, this is a review of our current understanding of the mechanisms of respiratory sensitization. It's going to summarize available models and approaches for their fitness of purpose, and address some regulatory needs, and have an eye towards the future projects to be developed. So if you think of a DRP as kind of like your preparation before you actually maybe define a new test guideline. Again, we're co-leading the project with the European Commission, and there's actually a lot of people also, I think, Austria, Luxembourg, the Netherlands, ICAPO. So the project there's been broad interest on this project ever since it was initiated and discussed. And, you know, I think we're all kind of excited that the project will help identify opportunities where we can improve our approaches to predict respiratory sensitization. The other project on here to mention, which again, I will discuss further later in this talk, is the revision of guidance document 34. So guidance document 34 is the foundational document for the test guideline program on the validation and international acceptance of new or updated test methods. We're co-leading this with the European Commission out of Netherlands. And so it's not that this document is not working. Really, it's an opportunity for us. The document is almost 20 years old. It was published 2005, and we have an opportunity to, you know, as any program that's been around for about 20 years, it's good to take a new look at that, see where it's been working well and where it hasn't, and then have a vision for looking forward. And I think what's kind of neat about this is, you know, remember this was published 2005, almost 20 years ago, as a frame of reference before the iPhone, right? So think of how much has happened since then. And so I think we're all very interested in taking a look at that. Next slide, please. So remember I told you there were a number of documents that were approved. So the previous slide, that's all the new work. And then looking forward, I just want to mention one of these in particular, where the US co-led the project with Japan, is the MIOGRT, or the Test Guideline 240 Madaka. And basically, you know, this was a revision of an existing test guideline, looking to extend the test to be more accurate or more sensitive to endocrine disrupting chemicals, and looking at the hatching cycle in the second generation of these fish. So just letting you get some context on what some of these titles are. Next slide, please. And on this slide, again, approved projects. The one that Tim will be talking about later, although I guess I can introduce it a little bit, is the initial guidance document on the evaluation of data from the Developmental Neurotoxicity In Vitro Testing Battery, the DNT IVB. And again, so what's interesting about this document is that it's describing different assays that comprise the battery in terms of neurodevelopment, and how we can assist in the determination of that certainty on positive or negative findings, right? Because there's a complexity in our nervous systems. And so what we're finding is that a battery is more helpful here in actually making a determination rather than just relying upon one single test. So that's what's kind of neat about this guidance document, and Tim will describe more about that in his talk. Next slide, please. So now we'll talk about some of the other updates so remember I said the WNT meeting is looking at new work that's coming in, the new proposals. We approve or complete some of these other projects, and these are some of the other activities which we discussed at the meeting. So the next slide, please. So ESCA is the Emerging Science and Chemical Assessment. It's an advisory group. So most of you probably won't interact with this, but what's interesting about this is an advisory group within the structure of the CBC. It's not a specific working party, but as an advisory group, you know, it's meant to serve as a resource to the other working parties, to advise on emerging scientific approaches where maybe, you know, the working parties individually don't have the expertise, and that's where ESCA will come to support and provide perspective, especially as we move forward with NAMs. There are so many different technologies that are coming where people just are not aware, or maybe don't have the depth of the knowledge or understanding. And so ASCA will help to develop guidance or recommendations on some of the emerging science and technologies for the benefit of regulatory application and for the members of the working parties. ASCA also oversees the components of the AOP program. And so of course, AOPs help describe that sequential chain of linked events at different levels and determining different levels of adverse health or eco-toxicological effects. And so we're excited that ASCA is formed, and I think we're all eager to see how it's going to help as we move forward in looking at emerging technologies. Next slide, please. In addition to the AOP program, the AmEx reporting framework, ORF, is another tool for which the OECD is committed to using tools to evaluate chemical safety. And so as a first step in looking at different effects of chemicals, how can you better collect data and standardize it in a way that it's more easily interpreted by decision makers to evaluate effects, and also as a platform to share information. So AmEx describes approaches to measure genes through transcriptomics, proteins, proteomics, or other small molecule metabolites. And then specifically for toxicology, these can help characterize or quantify where you have cell tissue or organismal changes following exposure to chemicals or toxic substances. And so the platform has been developed, the different frameworks are working together, and I would say that we're in this still conducting trials of the framework to confirm its utility. Is it doing what we hoped and improving the guidance document? And I know that we're very close to having that fully functional. Next slide, please. Test guideline program. So US is very engaged here with the planning and presenting through all phases of these activities. So we had an interest in 2022 to start looking at how can the WNT, the test guideline program, improve our ability to receive and review emerging technologies. Like we're seeing more and more products coming through at a faster pace. And so when you think about remember that process I showed you, even though that was an oversimplified diagram, you can still see there's a lot of steps in that process. And so as a way for us to help move that process along, we held a number of webinars in 2022 specifically to look at how we can improve WNT's ability. It covered a number of topics: validation, ecotoxicology, method readiness. All of those webinars are available on that website. It helped to build the foundation of knowledge, get everybody on the same page for the WNT coordinators to hold an internal workshop in December. So it's kind of like, let's do a self-evaluation, if you will. And again, continue the discussion on how to prepare a test guideline program for emerging technologies. We discussed, evaluating test method readiness. We talked about the concept of performance standards. We talked about non-standalone methods. We talked about how to develop guidance for batteries of assays. How can we incentivize validation? And then how can we better report validation study results? So those discussions are summarized, and there's a summary document behind that link. That helped to form the next piece, which was, remember I talked to you about the SPSF. So WNT, new work comes forward through a WNT SPSF that gets placed on a work plan. So those webinars and then the workshop help set the stage for the approval of the SPSF to work on Guidance Document 34, that foundational document for the test guideline program. And, you know, I think what's interesting is that Guidance Document 34 still works, it's still flexible, but, you know, how can we improve, or where do we need to improve our program to better receive the emerging technologies, right? How can we ensure that the robustness and certainty of a method before moving it forward for validation? Or how can we look at defined approaches for each information source, and then how do you look at them collectively, and then how do you validate them, right? So these are some of the, instead of a traditional like single chemical, single test endpoint. I think from the workshop, we had general agreement on the status of some of the issues related to like readiness and performance standards. We recognize they're complex topics that need to be scoped. It's going to take a lot of effort to move this forward. However, the work group is in place, of which we are co-lead. We're working on the draft revision as we speak, and I think it's going to be, I think we're trying to see how far can we get as an update on progress by our next meeting in April, recognizing that some issues will be more challenging than others. I will mention here one other thing that came out of the workshop, which is a recommendation for a validation workshop and a call for financial support. So next slide, please. So as a quick action, the Secretariat published, or released rather, in January 2023, a request for grant-making institutions and member countries to prioritize the evaluation and validation of emerging science and technologies. So really, how can we assist in accelerating the pace of chemical safety testing assessment through like NAMs, reduce the use of laboratory animals and better protect human health, funding, training, and also dedicated staff, right? So this is setting up a request that then leads to a workshop in December where, try to develop an understanding of validation stakeholder community, understand some of the needs and drivers, discuss and determine acceptable and good practices, those that we want to promote going forward. Again, look at some of these complex questions of determining robustness and certainty of a method before moving it forward for validation. What are some of the barriers that stand in the way? Or what are some of the other things that might be promoted that's working well? We're going to disseminate a survey in advance of the workshop so that we can learn different practical experiences with validation, maybe different aspects that need to evolve, or different opportunities for process efficiency gains, trying to get an understanding of the cost of validation, and maybe some of the things that help operationally make a successful or unsuccessful validation study. So that in the end of that, we can, again, have this gained perspective and then also identify future drivers and needs. Next slide, please. And so now we're back to, again, the oversimplified diagram for the test guideline process. So I'm hoping that what you heard or realized is how much work it takes to develop an idea, demonstrate repeatability and validity, have it reviewed by experts and approved by the WNT. We saw for proposal, how the new work is added to the WNT calendar. We heard about the extensive amount of attention the WNT is giving to improve validation for existing processes, and then ensure method readiness. Heard about the role of ESCO to support and advise new technologies and promote tools like AOPs and the AmEx reporting framework. And then, with the goal of the WNT to revise guidance document 34, with a focus toward how to guide validation and review of NAMs. And so, we're working across this process, with new tools and I guess you could say new internal policies to improve the test guideline review process and also better enable it to receive NAMs. Next slide, please. Just a few dates to place on your mental calendars, if you will. So those SPSF proposals are due to the Secretariat by November 15th. The stakeholder workshop will be held in December. And then the next WNT meeting is scheduled for April 2024. I think the next slide. Yep, last slide. So I recognize we'll have questions later after Tim speaks. And if you have any others, feel free to email me. Thanks.
Thank you, Charles, for that great presentation. As a reminder, like Charles said, we're going to hold all questions to the end, but please type them in as you have them using the GoToWebinar questions panel, and we will ask them after our next presentation. And again, please note if your question is for a specific speaker. Speaking next will be Dr. Tim Schaefer. Tim is a research toxicologist and principal investigator in the Biomolecular and Computational Toxicology Division, in the Center for Computational Toxicology and Exposure, in the EPA Office of Research and Development. Tim earned his PhD from the Department of Pharmacology and Toxicology at Michigan State University. And for the past decade, Tim's work has been directed towards developing new alternative methods for assessment of neurotoxicity and developmental neurotoxicity, hazards for chemicals. He has published over 100 peer-reviewed journal articles, book chapters, and reports, and has received 8 scientific and technological achievement awards and bronze and gold medals for outstanding service to the agency. In addition, Tim has held a number of different appointments and elected offices outside of EPA, including serving on grant review panels for NIEHS and in leadership roles for the Society of Toxicology's Neurotoxicology Specialty Section, as well as for other associations. Tim, thank you for being here today, and please go ahead.
Well, thank you, and thanks for the opportunity to present in the first presentation of this series, and so I'm looking forward to future presentations and EPIC meetings as well to hear what's going on in this area. So I'm going to talk today, as Charles mentioned, about the developmental neurotoxicology in vitro test battery, or the DNT IVB. And okay, my slides are advancing, good. So on April 26th, the National Committee of Test Guideline Program, or the Test Guideline Program, approved this document. It's already been mentioned. This is the guidance document on evaluation of data from the developmental neurotoxicity in vitro testing battery. And yes, that's a mouthful. And so this was a long time coming. I think it's a watershed event for developmental neurotoxicity testing because it symbolizes a certain level of readiness and acceptance of using data from these types of assays for environmental decision making. And so, as Charles said, this is a long process. This actually started a little bit even prior to 2003, and this is kind of what happened in the scientific field over that time. And that just kind of got us up to the point in about 2017, 2018, when we started talking about actually developing a guidance document. And so this is really the culmination of 2 decades worth of work. And so in an overview, I want to talk about why it's important to address developmental neurotoxicity, talk about the battery and the assays that are in it briefly. I really want to kind of present things in a context of how do we establish confidence the use of data from this battery, and especially regulatory use, because if the regulatory community doesn't have confidence in the data coming from the battery, then it's not going to be used, and it will be a lot of effort for naught. I want to give a couple of case study examples of how the data from the battery has already been used, and then the last slide I'll briefly touch on future directions. And so the public is obviously concerned about developmental neurotoxicity. Over the last several decades, there have been reports of increased neurodevelopmental disease in human populations. And there have been a number of epidemiological studies which associate exposure to environmental chemicals with increased rates of those neurodevelopmental diseases. And so the public is concerned about this, the agency is concerned about it, the global community is concerned about it. And here's where the problem comes in. So this is a slide I've used a number of times, but it really is a nice illustration. On the left-hand side here, we have about 10,000 different chemicals that are tracked by various different inventories that the EPA uses. So the IRIS database, toxic release inventory, pesticides, etc. On the right-hand side here are the number of those chemicals and a percentage that the EPA has some hazard data for. And so we have acute toxicity data for about 60% of these chemicals. If you come over here to the far right-hand side, we have developmental neurotoxicity guideline studies for less than 1% of those chemicals. Well, why is that? Well, the Guideline DNT study is a very slow and laborious process in and of itself. And it's also only triggered for pesticides, so it's not an absolute requirement for most chemicals. If a pesticide shows signs in other guideline studies of neurotoxicity, or if its structure is related to other neurotoxic compounds, then the agency will often request a DNT. But for other chemicals, for example, those regulated under TSCA, the agency does not typically request a guideline DNT study, and it's not required. One chemical to test it in a guideline DNT study costs about $1 million. It takes 2 years and uses about 1,000 animals. And at the current pace, since the guideline DNT study was approved in the late 90s, there have been about 200 chemicals tested in the last 25 years. And then the data from those studies has not been utilized widely, and that's because the guideline DNT study is a very complex study to conduct, and oftentimes the quality of data that come into the EPA or to other agencies is not as high quality as what might be desired. And so, again, starting back in 2003 and even before that, there was discussion of how do we do things differently? Can we do a better job? And the reason is because in the absence of that DNT hazard data, DNT doesn't get considered in making environmental decisions as an adverse outcome nearly as often as it perhaps should be. And so when you think about the development of the nervous system, it's a very complex process, and there are a lot of different things that have to take place at different times in different parts of the nervous system as it develops. And so that makes developing an in vitro approach to testing for neurotoxicity, developmental neurotoxicity, a challenge. But the answer to that has been to think about these processes that are key neurodevelopmental processes, such as proliferation of neuroprogenitor cells, their migration to the proper place where they need to be in the nervous system, their further differentiation into neurons and glia and other cell types in the nervous system, and then the neuroid outgrowth and synaptic connections and formation of functional networks all have to take place. In the context of that, apoptosis is also taking place, and neurons are getting pruned out as they don't make the proper connections and things like that. And so the field decided to focus on these key neurodevelopmental events and said, if we can develop assays that evaluate chemical effects on these key neurodevelopmental events, such as proliferation or migration, a chemical that disrupts that process, regardless of when or where in the nervous system that happens, could potentially be a neurodevelopmental toxicity hazard. And so shown here in the red asterisks are places where we already have assays developed and are part of the battery. And so it's shown in more detail here. There are about 17 different assays that comprise this developmental neurotoxicity battery. So there's a migration in human neural crest cells, neuroid outgrowth in peripheral and central neurons. These assays were developed by Marcel Leist at the University of Constance. You'll note that a lot of these assays are based on structural information and changes in structure in cell cultures. And again, structure is a major endpoint in vivo for developmental neurotoxicity. At the EPA, we've developed assays for proliferation of human neuroprogenitor cells, for neuroid outgrowth in both rat and human neurons, for synaptogenesis in rat neurons, apoptosis in human neuroprogenitor cells. And then the one functional assay that's part of this battery is this rat neural network formation assay, which is based on the measurement of electrophysiological changes using microelectrode arrays. And so a snapshot of an MEA output is shown here, where you're looking at the bursting and individual spiking of a number of different neurons. And this is a functional assay, and biologically, it makes up the underpinnings of what might be measured in an EEG recording. And so the very same biological processes that combine to give EEG recordings are what's being recorded in the MEAs. And finally, over here on the left-hand side, we have a series of assays that were developed by Ellen Fritsch's group at the Institute for Environmental Toxicology in Dusseldorf, Germany, that look at rat and glial and neuronal and oligodendrocyte migration, differentiation, morphology, oligodendrocyte differentiation, and neuroprogenitor cell proliferation. And so this is the battery of assays that comprise this DNT IVB that was approved in the guidance document. And so what I want to do is talk about how we establish confidence in the use of the data from these assays. And this is from a paper by Anna van der Zalm that was published last year, and it's this framework for establishing scientific confidence in NAMs. And it talks about what is the purpose that you're going to use the data for, how relevant is the assay to humans, what kind of technical characterization has been done, has it been independently reviewed, and then lastly, are the data transparent and is there a certain level of integrity with the data? And recently there's been a draft proposal from ICFAM on, again, acceptance of data for new approach methodologies. And it reflects very much this similar type of thought process. These are the important components to establishing confidence. And so one of the criteria, or several of the criteria for inclusion of assays into the battery are shown here. One is that these were deemed ready for use in screening and prioritization by several different studies that were published in the literature. The assays that were included all had to test a common set of chemicals, and that data needed to be analyzed using EPA's ToxCast pipeline and made available in the in vitro database from EPA. And so that ensures a certain level of data transparency and integrity. And finally, detailed methodological descriptions in ToxTemp format were also required for all the assays. And so these ToxTemp forms are 20 or so page documents that really delve into details about the technical characterization and the development of these assays, and also discuss how relevant they are to human biological processes. So many of these assays in the battery are based on human cells, not all of them, but even the ones that are based on human cells, for example, the MEA assay, are very relevant to human neurobiology, as I mentioned with the EEG recordings. So again, in terms of establishing confidence, all these assays have also been published in the peer-reviewed literature. And so again, these are the processes here that the assays measure. These are the names of the assays that you would see in various documents. And again, these are the peer-reviewed publications for each of these assays. In addition to that, I mentioned these studies earlier the assays were reviewed independently. And this was a paper by Anna Vol Price and colleagues where they establish a number of categories and parameters within each of these categories that you would want in an assay that you would use for regulatory purposes. They developed a scoring system for all of these different measures, and then they scored a number of different assays that were available at the time. And they scored them with respect to, are they in method development? How is the performance or replicability? Are they ready for screening? And the scores are shown here for the assays in the green boxes are the ones that were eventually incorporated into the DNT battery. So again, they've been reviewed by an independent group of scientists looking at the individual assays. Well, Charles mentioned the process for review of these guidance documents and test guidelines. Now, this is not a test guideline, it's a guidance document, but this is the review process for the guidance document. You know, it was accepted into the work plan in 2017. It was drafted. The expert group, so there's a DNT expert group that's sponsored by OECD. They commented on the document first. It was revised into a second draft, and then there were three rounds of comments and revisions from the WNT. And so this includes all the member states. I think Charles said there were about 38. And so each time we got comments on the document, there were literally hundreds of comments that needed to be addressed. That was finally approved in April, as I mentioned. I want to mention two other reviews of the battery in its entirety, and one was a scientific advisory panel conducted by the EPA in 2020. And again, this is an external advisory panel that the Office of Pesticides convened, and the topic of that particular panel meeting was the DNT in vitro battery. That happened in 2020, and then this paper just recently became available online. It's an independent review by an expert panel of, again, the DNT battery and assay test results. And so there have been three different independent bodies who have reviewed the DNT in vitro battery as a whole. And there's a consensus in all three of these reviews that, you know, the battery is ready for use in screening and prioritization decisions and weight of evidence decision-making types of decisions. Now, the last document that I mentioned didn't specifically state that, but if you read the document, they talk about using these data in the context of other in vivo data that may be available for environmental decision-making. Another thing that all three of these reviews pointed out was that they believe that this battery should be a living process that evolves as the science evolves. And so as new assays are developed or as we improve these assays, that information should be incorporated and used in decision-making processes. So in the context of as these reviews were going on in the laboratory, there was a lot of testing going on. And so there were a number of different sets of chemicals that were tested either by the assays in Europe, and the same set of chemicals, a subset of those was tested at EPA. The National Toxicology Program has sponsored testing of a phase 1 set of compounds in all the assays. That is about 115 compounds. And we've just started testing another set called the phase 2 set. EPA has tested some PFAS compounds, and EFSA is planning testing on about 100 different compounds that we hope is pending in the next year or so. And so this kind of gives you an idea of where all these different compound sets are in terms of putting them into ToxCast. I should say they focused early on testing DNT reference positive and negative chemicals. And again, that's to help build confidence, as well as testing chemicals with in vivo guideline studies that have been conducted. And then a number of chemicals of interest to different agencies have also been tested. So the question is, are we making progress? And you know, this problem is we can't test in vivo DNT quickly enough. And I didn't mention this, but this is about a $10 billion problem just to test all the compounds in this list. Here's where we are. You know, again, there are about 200 compounds that have been tested in vivo guideline studies. Currently, there are data for at least 81 compounds that are available in ToxCast in vitro database. We have another approximately 115 compounds that are pending and will probably be released next year early on. And then another 125 that are currently undergoing testing and will be then pipelined into in vitro database. And then there are at least 476 compounds that have been tested in at least one assay. And so all this testing has really taken place in the last 5 to 7 years. And so in that amount of time, we will have tested more chemicals and have data now for more chemicals than we have from the Guideline DNT study. And so I think we are making progress and speeding up the process of getting information, especially where we have no information. A more important question is, we're starting to generate data. Is that data being used to inform environmental decision making? And so I'm going to give you 2 quick case studies to show how that data is currently being used. The first is a screening and prioritization level study, and this looks at 160 different PFAS compounds. I think most people are probably familiar with these compounds. It's a very large set, literally of thousands of compounds. They're structurally diverse, but they all have these multiple fluorinations on a carbon backbone. Unfortunately, there's extremely little toxicological information on developmental neurotoxicity for the PFAS compounds, but there is some, and unfortunately, what evidence we have is conflicting. So if you look at the epidemiological literature, the few compounds that are looked at, or just looking at PFAS exposure more broadly, the epidemiology is quite equivocal. So one study will report a positive association, whereas another study won't. And there are a number of different studies, and it's really about 50-50. There's no consensus in the field. For in vivo studies in rodents, there have only been a few compounds tested, and there have been some neurodevelopmental effects reported in rodents and other animal studies. And so when the EPA was thinking about how it was going to approach PFAS, it decided that having some information on DNT would be valuable. And so what we did is we tested 160 different compounds in the EPA assays that are part of the DNT IVB. And these data were recently published in this paper that Kelly Carson's is the first author on. And what we found is that out of 160 compounds, only about a quarter of them had any activity at all. And even fewer of those were selective in that they had activity in the absence of or concentrations lower than we observed cytotoxicity. And so, the good news is there are a lot of compounds that didn't seem to have any biological activity towards processes that are important to neurodevelopment. And we can identify now the ones that do have biological activity, and we can use that information to make decisions about how we want to test compounds in the future, and which of these compounds we might take into a more complex system. So the second example that I want to talk about is a weight of evidence evaluation for pesticide registration. And the background on this is that the Office of Pesticides received notification from different registrants that they wanted to register L-glufosinate ammonium or L-glufosinate acid as herbicides. Well, racemic mixture D-L-glufosinate was already registered, and we had a Guideline DNT study that had been submitted to the agency that showed important neurodevelopmental effects in the absence of maternal toxicity. It was also known that the D-L-glufosinate is acutely neurotoxic and that it had in vitro effects on the network activity in the MEAs following acute exposure. And so the question is, do we have enough information from the Guideline DL study to inform decisions about the L isomers? And what the agency really needed was some comparative bioactivity data for the DL versus the L-glufosinate isomers. So, is this L isomer much more potent, for example, than the DL racemic mixture? And so we studied these in a couple of our DNT assays. And what we did is we compared the racemic mixture to the L-glufosinate ammonium and 2 different samples of the acid in an assay for Neuroid Outgrowth and in the microelectrode array assay. And here are the data for the Neuroid Outgrowth. Somewhat surprisingly, none of the glufosinate isomers or the mixture impacted the Neuroid Outgrowth. However, the internal controls for Neuroid Outgrowth performed as one would have expected. Now, before I talk about the results from the MEA assay, I want to point out that, as you can see, this activity is quite complex, and you can extract a number of different parameters that describe the formation of the network. And so in the heat map up above here, what I'm showing is along the bottom are the different parameters. And when we put these data into ToxCast, we fit them in both the down direction, which is on the left-hand side here, and the up direction, which is on the right-hand side. And so loperamide is our assay positive control, and loperamide decreases the activity. And what I'm showing in the green are a heat map for the different potency values for loperamide. And the yellow means there was no effect. And so what we can see here is our assay positive control worked. Our assay negative control, which is glyphosate, performed as we expected. And all the different glufosinate compounds had no effects, with the exception of one parameter that went in the up direction for one sample of the acid. And when we inspected the dose response curve for this and also noted that there was no response for our other sample, we found that these data were not really convincing, and so we dismissed this as not being biological relevant. And so we passed this information on then to the Office of Pesticides, and I'm summarizing it here. There were similar effects of the D and the L isoforms in all the in vitro assays, and I didn't show was that we repeated with the acute assay what we had shown in 2014 for the acute effect on the networks. We did the in vivo to in vivo extrapolations and showed that our tested concentrations exceeded the points of departure for the L-glufosinate risk assessment based on other endpoints. And then again, the in vivo evidence again showed that there was similar toxicity profiles for both the DL and the L-glufosinate. And on the basis of all this evidence, because this is a weight of evidence decision, the agency granted waivers for the L-glufosinate, acid, and ammonia. And this slide really shows the impacts of that decision. So this is the timeline for making decisions based on the in vitro data. We started in March of 2019 when Office of Pesticides officially requested Office of Research and Development to collect the data. The decision was made in June of 2021. If at that point in time in 2019 of DNT guideline studies had been requested, it would have taken 3 years for the agency to get that data and another half a year at least to make decisions based on that data. And so we saved at least a year and maybe closer to a year and a quarter or a year and a half by doing the in vitro studies. It also didn't use a significant number of animals, so we used about 20 animals total for the in vitro studies, where it would have been about 2,000 animals for 2 guideline DNT studies. And again, the costs were about $100,000 for the in vitro study compared to about $2 million for 2 compounds in a guideline DNT. So across the board in terms of cost, animals used, and time, incorporating the in vitro data allowed us to do things much more quickly for fewer animals and much less expensively. I want to just point out there are other case studies that are available on the OECD website. So I've just shown you 2 of them, but there are several others if you want to check them out. And then last, I want to just close with a couple comments about future directions. So in those 3 reviews of the guideline or the battery itself, it was noted that some developmental processes are not well covered, particularly microglia, the neurovascular unit, and plasticity. There's work going on in all of these areas to develop and improve assays, develop new assays looking at microglial function. The interlaboratory transferability of these assays has not really been demonstrated. That's also a very active area of research where EPA, as well as the other laboratories that develop these assays, are working very hard to develop or to demonstrate the transferability from one lab to another. And then there's not a tiered testing strategy at this point in time. And so, again, that's something that EPA, as well as the other other players in this field are working on developing tiered tests, tests in three-dimensional system, tests in alternative species models such as zebrafish that I think will be coming in the future. And so with that, I just want to summarize everything. I hopefully have talked about how we have developed this battery of in vitro NAMs over the last 15 years or so. There's a consensus that we can use data from that battery for screening and prioritization and weight of evidence decisions. And I showed you a couple of example case studies for how the data from those NAMs has been used for that context. And I hope I've convinced you that we can speed decision-making processes, reduce costs, and still contribute to health protective decisions and provide information where there is no information using the DNT NAMs. And finally, that I think an important point is that there's consensus that the science behind the DNT NAMs are going to continue to evolve and improve. And so with that, I just want to thank all of my colleagues. This is not an effort that I have been the sole person on, obviously, and a number of people at EPA, both in our Office of Research and Development as well as in our program offices, have contributed to this. The heads of the laboratories at IUF and the University of Constance, Ellen and Marcel, also have teams of people working on this. And then Andrea Tarone and Iris Magnus at EFSA have also been involved, as well as Magda Sachana at the OECD. And so with that, I would be happy to stop and take questions.
Great. Well, thank you so much, Tim and Charles. Both of your presentations were really information-packed, and I know that's what people came here for. So thanks very much for these updates. It's always good, I find, being engaged in OECD so much, you kind of forget that people don't follow it as much as those of us who are who attend the meetings do. And so having you come and tell people what the latest is, I think is really, really important. So thanks. So we have a lot of questions coming in already. Everyone, if you have a question for either of the speakers, please feel free to put them in the question box and we will get to as many as we can. The first one is for Charles, actually. It's a question about ESCA and EGMIST, and it's how does ESCA relate to the activities of EGMIST? Does the first oversee the second?
Great question. I specifically didn't use the term of EGMIST because I know that we're moving forward. EGMIST was the previous iteration of ESCA, and what the Secretariat realized is that the activities of ESCA had really grown, and so there was a way to, I guess, break it back down and refocus its efforts to really be an advisory group on emerging technologies. So in a way, ESCA replaced EGMIST. So that's, I guess, the best way to think about it simplistically, and that the way it operates is different too now, because you remember the diagram I had with arrows that went back and forth between different working parties, is that ESCA now is really working in collaboration with working parties, where before I think it was working a little bit more self-driven. So those would be really the differences. But ESCA is the advisory group going forward, replacing EGMIST.
Okay, thank you.
Okay, next I have a really long question that I'm going to read out, and this is for you, Tim. Okay, if DNT is really such a big concern, shouldn't we have seen more dominant effects in the population over the last decades? In principle, he agrees that an easy to conduct battery testing makes sense for chemicals. However, I'm not convinced that a vast amount of the currently used and not tested chemicals really have this inherent potential. Another question is also whether there are really more DNT-related diseases in humans, or whether we have rather set a bigger focus on these diseases during the last years. Has there been any comparison between the research regarding this topic now and maybe 10 or 20 years ago?
Wow, yeah, that is a long question and a good question. And yeah, I think, you know, the comment that, and so let me try and break it down and remind me, Bridget, if I don't address something. So there are thousands and thousands and thousands of compounds out there that have not been evaluated for DNT. And yeah, from a kind of common sense proposal, you would think that if something were really, really nasty, that we would see real clear evidence of that in human populations. The problem is the lack of information, the lack of any ability to understand whether or not a chemical might have a hazard for DNT and at what level that hazard may take place. And when the agency is making decisions about, you know, how it's going to clean up a toxic site, for example, or, you know, what levels of pesticides might be allowable in food residues or something like that, if we have no information at all, then we can't assure any level of confidence that a chemical is either not causing or understanding what level it does cause. And so maybe we don't need to do a guideline DNT study on all 10,000 of chemicals that are in that list of chemicals that I showed, but I think most people would agree that it would be nice to have some information about the biological activity towards processes that are important in neurodevelopment. And you know, if we understand that, you know, the exposure levels are far above where we might see any biological activity, I'm sorry, far below, excuse me, then I think that gives everybody involved, and the public especially, a certain level of confidence and reassurance. And it helps us focus on where we might, you know, actually, usually an epidemiological study, there are a lot of chemicals that people are exposed to. And so understanding why we might be seeing neurodevelopmental effects, you know, and where to focus our efforts is also valuable. And so if we can even just rule things out, I think there's a lot of value in that. And again, yeah, I think the other part of the question was, you know, are the neurodevelopmental diseases actually increasing, or is this just, you know, a result of being more focused? I have to admit, I'm not an expert in this area, but the literature that I've read, there is a certain level of understanding and looking for these diseases that contributes to the increase. But I don't think, in what I've read, I don't think we can entirely explain away some of these increases entirely on the basis of increased diagnosis or something like that. So was there another part of that I didn't address, because it was a pretty complex question?
I think you hit all of it, Tim.
Okay.
Yeah, I think you got most of it. Here's a question about validation of the DNT in vitro battery. Specifically, they're wondering whether organics and inorganics were used, and they're kind of highlighting the issue that sometimes, or often, I guess, a lot of new methods are validated quite skewed towards organics.
Right. And so I mentioned the testing of DNT reference chemicals. And so a number of years ago, there have been a couple of publications, one by Bill Mundy, one that Mickey Ashner is the first author on, that suggested different sets of chemicals to evaluate and look at the sensitivity and specificity of in vitro assays for developmental neurotox. Those chemicals consisted of ones where there was some level of evidence that the chemical caused developmental neurotoxicity in a mammalian species or even in humans. Unfortunately, we don't have a huge database of chemicals to draw from here, but those chemicals included heavy metals like lead and methylmercury, tin compounds, so inorganic compounds, as well as some pesticides, as well as a number of different drugs where neurodevelopmental toxicity has been, reported in mammals or humans. So it wasn't a strictly limited set to organic chemicals. In terms of chemical space, though, it could be broader. But the problem is we don't have data for broader classes of chemicals on their developmental neurotoxicity in vivo. And so it's more challenging to come up with kind of standards, if you will, for comparing in vitro to in vivo results. There's also a challenge in terms of identifying chemicals that are negative in vivo. And so that set is smaller. Now, as we continue to test more chemicals in this DNT battery, the chemical space that we're testing is getting larger. And so we'll know if chemicals at least can be tested in these assays and give us biologically relevant responses.
Right.
Okay, there's a lot of questions for you, Tim. The next one is, what assays were used to evaluate the 160 PFAS compounds?
Okay, it was mostly the EPA assays. It was the network formation assay, proliferation, apoptosis, Neuroid outgrowth in both a human cell model and a rat cell model. I think the only assay that we didn't have data for was the synaptogenesis assay. But there's a lot of overlap between the network formation assay and the synaptogenesis assay. And so I think I've covered all the assays, but again, consult the paper for the specifics.
Thanks, Tim. I will mention there are also some compliments coming in. Thanks to both of you for clear presentations that answered some questions already. So with that said, there's a question that I'm not 100% sure I understand. I think what they're asking is there a consensus on grading the PFAS compounds in some grading system? I think what they mean is some kind of binning or prioritization system. And so what are the strategies to translate the in vitro data to something relevant to human risk?
So I'll do my best to answer that, and I don't think I have a good answer, to be honest. I can tell you kind of, we collected the data from the developmental neurotoxicity assays. We did, and this has not been made public yet. It wasn't part of the publication because I'm not sure exactly what they're going to do with this, but we developed some flags for, this is an active chemical, this is a chemical that's active and not cytotoxic. As part of that larger project at EPA, there were a number of other in vitro NAMs type of assays that PFAS were being tested in. And so the current status of that is all of that data are being pulled together now. And there should be a publication upcoming in the next 6 months to a year that is a higher level summary of those data. And so that's currently being worked on, and I'm not a lead on that, so I can't really speak to where it is at this particular time, but I do know that it's ongoing, and that will help then guide future decisions that the agency is going to make about PFAS and future testing of PFAS. So I'm sorry if I can't completely answer that, but I'll at least give an idea of where it is.
It's definitely a process.
Okay, I have another question for Tim. I'll read this one out as well. Dobrzynski et al. Used QIVIVE to estimate fetal brain concentration and put into weight of evidence findings from the in vitro DNT battery. This approach comes with uncertainties. Were these quantified? Would an actual measurement of fetal brain concentration at GD20 be appropriate where these studies are generated anyway for registration, such as for pesticides?
So I think there's a minor correction there. I think the IVE there did not estimate fetal brain concentration. It estimated blood concentrations and estimated really doses that would give rise to the blood concentrations that were equivalent to what we used in vitro. Those uncertainties were all taken into account. You know, this went through a fairly rigorous review process by two different committees at the Office of Pesticides that are involved in the granting of these waivers. And again, that's getting a little bit outside my area. You know, the Office of Pesticides ultimately did the review and the weight of evidence decision making, but it was reviewed pretty thoroughly, and so I think the uncertainties were considered. You know, they had other studies from the L-glufosinate that indicated much lower points of departure, I think, and so that's why they ultimately used those.
Thanks, Tim. Okay, here's an interesting question about a couple of questions about comparing to humans, and I know you addressed this somewhat in your presentation already, and I know that's top of mind for the group. They're asking about biomarkers for DNT in humans, and I'm afraid I don't know, or maybe you do, if there are any that have been established.
Not that I'm aware of a specific biomarker, and again, you know, DNT in humans it covers a wide variety of things. It can be structural alterations, it can be behavioral alterations, it can be learning and memory. So there are a lot of different potential endpoints. And so going back in history to when this field was first getting off the ground, there were a lot of discussions about, you know, should we develop assays against, for example, this kinase, because we know it's really important in neurodevelopment, or should we be developing something that is higher order? And ultimately, the consensus was reached that, you know, these neurodevelopmental processes are the things where we need to target assays for, and for two reasons. One was, if you use a target-based approach, like, you know, this enzyme or that ion channel or this receptor, there's literally thousands of those in the nervous system that could be potential targets of environmental toxicants. And we don't know where and when to look. The other thing is, those processes are fairly well conserved, and disruption of those processes can pose a hazard for the developing brain. And so if you don't have proper proliferation and form enough cells to form the nervous system, you're going to have problems. If you don't make the right synaptic connections, if you can't form the networks that are important to, you know, different processes in the nervous system, again, that's a potential hazard for neurodevelopment. And so that's the reason the key process approach was taken and not a more target-based screening approach. So I hope that kind of addresses that question. It's a difficult one because it would be nice to have biomarkers that, you know, you could assay, and that would, I think, certainly aid epidemiology if you had specific biomarkers, because behaviors are difficult to assess, especially in human populations.
So maybe just to jump off of that point really briefly, excuse me, Bridget, but this one follows on a little bit, which is, given the complexity of this whole area, someone is asking, how many assays do you envision? I know it's not just you, but are envisioned being in the DNT battery? Specifically, they're asking for companies seeking regulatory approval.
Right, and so right now, you know, the battery is at about 17 assays. In order for other assays to be added to the battery, they would need to go through a similar process where, you know, they tested reference chemicals and, you know, we're willing to share that data with the agency and put it into ToxCast and to to write the ToxTemp forms. And so, it's not going to be, I think, a quick process to add a new assay to the battery. And as Charles pointed out, these assays are not test guidelines, and so that process may need to happen in the future for these assays as well. We're at a point where there's guidance, and so that means agencies could consider data from these assays. The EPA has the ability to consider data from any number of different in vitro assays as they make decisions. So, again, depending on the context of the decision, the agency making the decision, and what the legalities around the decisions are, different types of data could be considered. In terms of this battery, right now it's at 17 assays. In the future, there are multiple, for example, Neuroid Outgrowth assays. We may decide that, the value of information from all those assays is not sufficient, where we only need 1 or 2 of them. And so that number could also go down in the future. We're early on in the use of this battery. It's now at the point where there's a certain level of acceptance, as indicated by the OECD guidance document. But I think we're still going to learn more about these assays, and as I mentioned, the science is going to evolve, and so it's hard for me to predict, and maybe that's why I'm, how many assays are ultimately going to be needed, or how many are ultimately going to be incorporated. It could change in either direction.
Yeah. And it maybe depends on the context of use, or what we know about the chemical already.
Yes, that's an excellent point, Christy. Actually, I'm glad you brought it up, because I didn't really talk about one aspect of that, and that is that these assays are intended to be used in what's known as an integrated approach to testing and assessment. And that really is the glufosinate study. We looked at what information we had and said, what information do we need and how can we get that? We didn't run the entire EPA battery. We ran 2 assays because we thought that would give us sufficient information to make the decisions that we needed to make. And so, in that context, the question may be asked, well, what decision are we making? And so we may not run the entire battery. We may only need data from some of the assays.
Yeah.
Okay, we have a lot more questions. Probably not going to get to all of them, but so trying to pick and choose and prioritize a few. But this one's again for you, Tim. Are there test methods designed or under the pipeline for chemical transport across placenta and blood-brain barrier?
Yes, and I didn't have time to talk about those kind of assays, but there is a fair amount of work going on at EPA as well as other places on especially blood-brain barrier transport, and really the work on the placental barriers, I think, is a little bit more theoretical rather than developing models for transport of chemicals across the placenta. But there is work going on in those areas as well. I just didn't have time today to go into that.
Okay, thank you.
Another interesting question is, how does DNT differ from general neurotoxicity when it comes to in vitro assays?
Okay, yeah, and that was something I touched on very briefly that's in the guidance document. And so one of the things that was outlined in the guidance document is, we need these assays to be relevant to the developing nervous system. And so maybe the best example is with the MEAs. I can take neurons, plate them on the MEAs, and let them develop neural networks for 2 or 3 weeks, and then expose them to chemicals. Well, if I do that, by that time, most of the network development has happened, and you're not looking at a developmental process. By contrast, the network formation assay that is part of the battery, what we do is we plate the neurons on the MEAs, give them time to attach, and then expose them to chemicals as those networks develop. And so now we have an exposure that's relevant to the developing nervous system, whereas in the other example, that exposure is really an acute exposure that measures neurotoxicity and doesn't have a developmental component to it. And so all of these assays expose the chemicals to the system during a process that's important to neurodevelopment. And so that was kind of the distinction that's made. You know, you can take a PC12 cell and expose it to chemicals and look at neurotransmitter release. But if you're only doing that for a short period of time, you're not really looking at a developmental process. So hopefully that clarifies that a little bit.
Is it my turn for a question, Steve? Okay. All right. These are more for you, Tim. Regarding the in vitro assays for DNT, there's no mention of controls for metabolic activation of chemicals, as often seen with genotoxic chemicals. Are there known developmental neurotoxicants that are activated by metabolism by P450s, et cetera?
Yes. And this is an area where, again, there's active research going on at the EPA. For those chemicals that we know are metabolically activated, actually a good example is the organophosphates, right? Many of the parent chemicals themselves are not the insecticide, but they're metabolically activated to oxon forms. And so we have done a lot of testing of organophosphates, both the parent and the oxon. And so when we know metabolites exist, we can easily test those metabolites in the assays that we have. I think the bigger challenge is for chemicals where we don't know much about their metabolism. You know, there is a differing level of metabolic capability in all of the model systems that we use here. And so it's not to say that no metabolism would occur, but it may not be the same metabolism that you might get in an in vivo exposure where, you know, it passes through the liver and it's activated by the liver to a neurotoxic compound. There is work going on at EPA as well as other places to add metabolic competency to these types of assays. And so in the future, hopefully that science will be something that we can incorporate into these assays.
Well, I think that's all the time we have. I really appreciate both of you for your presentations and for your time answering all of these questions. To the audience, thank you so much for joining us. In fact, you're in good company. We had over 900 people registered for the webinar, and we had nearly 400 people on the line today. So thanks so much for such a great response to this first EPIC webinar. And I'm sure it was partly thanks to you, Charles and Tim, for having some attractive presentation material to share with us. As a reminder, there's going to be the slides and the recording will be posted on the PETA Science Consortium website, and that is in the chat in case you don't know where to find that, but you can also just Google that as well. Okay, well, thanks again also to our hosts from EPA and California EPA. Very excited to kick off this new series, and I hope you all have a great rest of your week. Take care.
Bye, everybody.
Thank you. Bye-bye.
Bye.